Before I begin, I really want to thank the organizers for having me here today. It's so nice to be out of the cold and snow of New Hampshire and into the relative warmth of Paris. My wife and I have had a fabulous time. It's just been a wonderful experience. And I'm very grateful to be able to speak with you today on what I really love is the Cambrian explosion of animals. And so before I begin, I just want to acknowledge NASA that funds my research and my colleagues here who have always made my kind of bad ideas at least somewhat better just through vetting with them. So this remarkable painting is what the ocean might have looked like about 505 million years ago in the Middle Cambrian. And you have this veritable potpourri of just strangeness. If you're an invertebrate zoologist, you might be able to recognize a few things, but this is a pretty bizarre group of critters. This here is the giant beast of the Middle Cambrian called the Anomalocaris. It's about 1 meter long. It's as big as life got in the Middle Cambrian. And you can see here it's terrorizing some arthropods here. This is actually thought to be related to potentially modern spiders. Shown here are sponges. They pretty much look the same. Sponges don't do anything for 600 million years. It's the most boring thing on the planet, but and I work on them, okay? So it tells you a lot about me. This thing here was named by Simon Conway Morris Hallucigenia. It's a good name. Pink Floyd, LSD-inspired name. This here is us. That's Pikaia the chordate, and so that's a relative of us. We're members of the phylum Chordata as vertebrates. So if we jump ahead now 505 million years, this is where you would find these fossils. This is the Burgess Shale up in Yoho National Park in British Columbia, Canada. And it's just the black you see there. So if you go off on either side into the white rock you see, you actually won't find any fossils. It's only that black outcrop. And it's a slump where all the animals basically got caught in a landslide and were carried down to the bottom of the ocean and then were buried. And so here's what these fossils look like. That's an Anomalocaris. Just a spectacular fossil. And this is my brother and I. We were fortunate enough to be able to go up for the 100th anniversary of the discovery of the Burgess Shale. And so we walked, it was about 500 miles, I think. It was a really long ways. I don't know how many kilometers that is. It was a lot. We were very tired. And within, of course, the rock is just all these wonderful fossils. Now, if we think about the fossil record, the Burgess Shale is a spectacular deposit of soft-bodied forms, okay? There's actually not that much skeleton in the Burgess Shale, right? It's soft-bodied stuff, so you can see guts and muscles and all these animals that basically you don't see anywhere else in the world because, of course, they don't preserve. But we've known about the Cambrian fossil record, of course, since before even mid-Victorian times. Darwin spends a tremendous amount of time in Chapter 9 of The Origin because, of course, it's the biggest problem he faces in terms of trying to convince people that evolution is a reality. There is another and allied difficulty, which is much graver. I allude to the manner in which numbers of species of the same group, what we call a phylum, an arthropod, a mollusk, a vertebrate, what have you, suddenly appear in the lowest known fossiliferous rocks. Because the fossil record is exactly what a creationist would predict. You go from nothing to everything in the geological blink of an eye. And Darwin's trying to convince his readers that actually there's all this time where animals were there and evolving. So there has to be a way of getting around this problem. He's speaking of, in terms of thinking about this explosion of fossils, work derived from Roderick Murchison, one of the greats of geology. And he wrote a very famous volume called The Silurian System, and this is one of the plates describing some trilobites from Wales. Now, this is the problem. That's the fossil record of animals, but animals are basically a proxy for pretty much all forms of life, at least especially as we understood it in mid-Victorian times. Shown at two hierarchical levels, phyla, arthropods, mollusks, vertebrates, etc., or classes, whether crustacean, insect, chelicerates, that sort of thing. Okay? This is the Precambrian. That's the Phanerozoic. Nothing to everything, boom, in the Cambrian. Okay? We've known about this pattern since Murchison published the Silurian System. And all we've done over the last 150 years is just basically add more and more data showing the robustness of the pattern, making the problem more and more and more difficult. Darwin predicted that we should be able to find all these ancestors. We published this paper in 2011. Where the hell are they? Now, I'm going to be talking about animals primarily. I thought that that was kind of what I was supposed to do, but I wanted to point out that animals obviously are not the only things that form skeletons. In fact, this is one of the earliest skeletons known in the fossil record, described by Phoebe Cohn and her colleagues. It's a eukaryotic protist of some sort. We have no idea what kind of protist. We don't know what it's really related to in terms of what it's living today. It's about 750 million years old, give or take. It's phosphatic. Okay? But in terms of thinking about the fossil record, what we think about when you go to the Natural History Museum, what are we seeing in that hall of invertebrates is, of course, that dramatic rise throughout the last 500 million years. So let's take a closer look then at what we mean by this Cambrian explosion. What we've got here is a series of animals. Okay, who can name that animal right there? That's Stephen Jay Gould, one of the paleontological greats of the 20th century, at least in my very humble opinion. I love Steve. I think he's fabulous. And of course, he wrote extensively on the Cambrian explosion, and I got into this because of his book, Wonderful Life. If you need a good airplane read, fabulous book. Okay? Now, again, here is the fossil record shown here in blue and yellow. Time here is shown on the left, starting at 800 million years, moving through the snowball Earth events, leading up into the Precambrian-Cambrian boundary right here, up into the end of what we call the Ordovician. Now, shown in black are the fossil records derived from skeletons of those particular animals. Okay? Shown in gray are fossils from those particular animals, like hemichordates, that don't have hard parts, but are known from places like the Burgess Shale. So you go to the Burgess Shale, you actually find hemichordates, but you don't have skeletons of hemichordates in the same way that you do, say, echinoderms. Okay? Here's vertebrates that we'll be hearing about after me from Philippe. We have some soft-bodied vertebrates we know right in here, and then the hard part record kicks in somewhere in the middle Cambrian. Now, we're going to be talking a little bit about taxonomy. I apologize for the words. They're terrible. I have no control over this, okay? The deuterostomes, the second mouths, which includes us, okay? So there we are, the vertebrates. Our nearest relatives are the echinoderms and the hemichordates, okay? And that is a fossil deuterostome. Some have described this as a vertebrate, huh, Philippe? It's actually an echinoderm made of sterium, okay? It's a carpoid. It looks nothing like a living echinoderm. How many arms does a starfish have? 5. Echinoderms are 5-fold symmetrical. This is totally asymmetrical. It has no water vascular system, so it isn't actually pumping any water. It's got a tail. I think Dick was right, and it probably does have gill slits, which is a primitive feature for deuterostomes, but it looks nothing like a living echinoderm except its skeleton. It's unequivocally an echinoderm. Nobody else makes sterium. Now, the next major group is what we call the Lophotrochozoa. Because some of these animals have lophophores, like brachiopods, shown here as an early Cambrian. They show up about 520, 25 million years. And the trochozoans are things like mollusks and annelids because they have trochophore larvae. So you take the lophophore and the trochophore and you combine it together into a god-awful name. Again, fossil records in black. It's possible that the mollusks go into the Precambrian. I'm not going to talk too much about that, but that's what the white bars indicate, is that these are possible range extensions. So sponges may go back into the Cryogenian, all right, long before skeletons of sponges actually show up. Mollusks may go back into the late Precambrian. The last group is called the ecdysozoans because these animals have cuticles that they shed. In other words, they undergo ecdysis, and includes things like arthropods, nice little trilobite there, preapulids, the penis worms, and onychophorans, the velvet worms, okay? As well as things that basically have no fossil record like nematodes, okay? Now, preceding the Phanerozoic, the Cambrian explosion, if you will, is what we show here in green, is what we call the ediacaran fauna, okay? Now these are a group of critters, and I use the technical word there, critter, which basically tells you we have no idea what the hell these things are, okay? You can find people who will tell you what they are, okay? But that doesn't mean we know what they are. I think this thing is probably related to modern placozoans. Bruce Runiger would probably say maybe that they're annelids. People have described them as fungi, as cnidarian. You name it, you can put this thing in it, okay? And this animal, organism, critter, fungus is called Dickinsonia, and it's one of numerous critters that fall within the latest Precambrian fossil record. What's so strange about what Darwin predicted is that, okay, we found fossils. Right? You read Origin, like, hey, God, I could really make a name for myself if I can go and find Darwin's ancestors. And you go to the late Precambrian, and you find stuff. What the hell is it? Right? It's really, really hard to relate that Ediacaran with what we find in the Cambrian. It appears like you're just on two different planets. And the boundary, it's fast. This thing is only a few million years old before the Precambrian-Cambrian boundary. And then, bam, in the blink of an eye, it changes. Now, there are a couple of interesting things there that show up at the very latest Precambrian, okay? And what we call the Nama assemblage, which is named after a formation in Namibia. Cloudina and Namacolothus. These are biomineralized hard parts of what are assumed to be animals, okay? And I stress the assumption. They are assumed to be animals. Some people would describe this as an annelid because modern Pogonophorine worms make tubes, like cone in cone, like Cloudina. But they don't make it the same way. Namacolothus, what do you guys think it is? What do you think? Huh? Sounds fabulous. Why not, right? Why not? No idea. So they're carbonate, they're skeletons. They may be animals. They're assumed by most paleontologists to be the products of animals. In fact, what people say is that it's probably a sponge or a cnidarian because they're like the simplest thing. Even though they have nothing in common, sponges and cnidarians. You might say it's an elephant or a chimpanzee. You know, what the hell? They're all the same. This is the beginnings of biomineralization and skeletonization in the animal fossil record, assuming that these are animals. Okay? They may not be. Now, what I'm going to show you here now is let's move our way now. That's basically the Precambrian fossil record of animals right there, okay? Again, assuming they're animals. We're going to move into the Cambrian. Now, it's a little complicated, this graph here. Let's take a look at it. We're going to split it up by taxa. You got sponges over here. What they're called cnidariomorphs. Things that are related to modern sea anemones and corals and jellyfish and the like, okay? But would include things like this because it looks like it's simple enough to maybe be a cnidarian, even though it has no characters or real similarities with living cnidarians. Okay, so we're going to call them a cnidaromorph. We're going to have a group of animals here called chaetognaths. There's our Lophotrochozoans, our ecdysozoans, and our deuterostomes. Moving this way then is we're moving towards the recent. So we're older here, moving to 510, and then of course 0 would be way up there. Okay. Here's Cloudina. Our key here is the mineralogy. So you can see here is that Cloudina is made of aragonite. Okay. So it has an aragonite skeleton. Other things like Namacolothus have high magnesium calcite skeletons. So the first things that show up are either aragonite skeletons or skeletons made of calcite, but with high magnesium. Okay. These are the sponges. Now sponges, demosponges in particular, make their skeletons out of glass. They use silica. Okay? And spicules show up sometime in the early Cambrian, but sponges themselves appear to show up in the Ediacaran. Okay? So these arrows going downward, people have described sponge spicules from the Precambrian, but they all look like garbage. Real sponge spicules made of glass show up in the Cambrian. But there are things in the fossil record, I've even described one of them, that will appear to be architecturally a sponge but does not have spicules. Okay? We have another group of sponges here called the calcareans, and then another group called the archaeocyas. Their skeletons, high magnesium calcite, they don't show up until later in the fossil record. Okay? This is the protoconodont. Okay? These skeletons are made of phosphate, right? Just like our skeletons are. Right? These are hydroxyapatite skeletons. And I want to make sure we're clear on this because it can be confusing. I think Philippe will probably talk about conodonts as well. Conodonts are polyphyletic. When people were first working on these things, they thought, well, maybe they're all one suite of evolutionary grades, such that these simple conodonts evolved into these more complex conodonts. It appears that that's false. These are actually related to modern chaetognates. We call them arrowworms. They form a tremendous amount of the biomass of the oceans. They eat small crustaceans and then are eaten by bigger fish. So they're a key link in the marine food chain. These are related to vertebrates. So they have nothing to do with one another. So when Philippe is talking about conodonts, he's talking about these. When we talk about conodonts, we're going to be talking about the chaetognate grasping spines. I won't mention these again. We move into the Lophotrochozoans. This is a beautiful fossil called a hyolith. Right? Again here, it's an aragonitic skeleton. In fact, you see that mollusks, hyoliths, halkyarids, waxids, etc., are all making their skeletons here out of aragonite. And hyoliths constitute a small part of what we call the small shelly fauna. Now, if you're a Cambrian worker, especially in the very earliest Cambrian, what you do is you take rock and you macerate it and you dissolve it in a weak acid solution. And what you leave behind is a whole suite of phosphatic fossils. Right? That look like this. Now, some of these were originally phosphatic, but most were originally aragonitic, and they've been replaced with phosphate. So I can then use acid to dissolve them out and to study them. Now, these are bits and pieces, for the most part, of various animals. In fact, this one here is called Microdictyon. It's a beautiful little thing, isn't it? It looks like it would be a nice piece of jewelry if it was like a little medallion or something. I think it's wonderful. But Microdictyon is actually related to this animal here, Hallucigenia. And what it is, it's the shoulder blades, if you will, okay? So every time they have a limb, they had a little patch of aragonite right there. And you can see these running down the body. There's an artist's reconstruction. And again, it's an onychophoran, like a modern velvet worm, just like Hallucigenia is. So when we think about these small shellies, right, these are all, for the most part, just bits and pieces of skeletons that have broken apart, okay? Here's the fossil record then of these small shellies. What you're looking at here is the cumulative first appearances of small shelly fossils. What we mean by that is that this is everything that's living today plus everything that came before it. Okay? It's not standing diversity. It's not like there's getting more and more animals. We're just getting more and more records as we move through time. Okay? Cumulative curve, aragonitic in blue, phosphatic in green, calcitic in red. Lots of different things. People like Stephen Bankston, I don't know if you guys know who Stephen is, one of the world's experts, describes all of these sorts of things. Now, as we move later in time, we see something interesting happen. We go from high magnesium calcite, or aragonite, to low magnesium calcite in terms of the skeletons that we find. So trilobite skeletons, shown here, low magnesium calcite, show up slightly later in time than these aragonitic forms, things like the hyoliths, like I showed you. And echinoderms, high magnesium calcite. This is what we call a Gogia. How many arms do you see there? 5. It's got stereome, and we've also got 5 arms. Okay, so this is phylogenetically more closely related to living echinoderms because we've got a water vascular system with 5 arms. Now, this split, it's pretty an interesting discovery, primarily made by Susanna Porter back in 2007. She's a co-author on this Maloof et al. Paper. Notice is that if you look at seawater composition during the earliest Cambrian period, it's basically going from an aragonite to an aragonitic calcitic to a calcitic sea. So in other words, animals that evolved their skeletons during what we used to call the Nemekit-Daldinian were living in an aragonitic ocean, an ocean that has high magnesium ratios, and we're using that particular mineral that they're finding in the ambient seawater to make their skeleton. But as we move into what they call the Timonian, we change over to a calcite sea, and animals that evolve their skeletal rings during this period or later use low magnesium calcite. So shown here then is seawater chemistry moving through the Cambrian. We've got silica. We have phosphate here. Okay. We've got aragonite and high magnesium calcite going down and low magnesium calcite going up, resulting in almost a dichotomy then in terms of the types of skeletons you find. Those that evolve skeletons early in the Cambrian primarily have aragonite or high magnesium calcite. Those that evolve later in time have low magnesium calcite. What's interesting about this then is that you can actually test for missing fossil records by looking at primary compositions of skeletons. If you find an aragonitic skeleton, say from the early Ordovician, one would hypothesize, I bet I'm missing fossils. I bet this fossil record goes back to the early Cambrian because they're using aragonite and not calcite. The other interesting thing that allows us to do this is that the ratio of seawater then determines the skeletal mineralogy, but then the animal keeps that mineralogy irrespective of what happens with the water around it. So if I evolve in an aragonitic sea, I have an aragonitic skeleton. I don't switch over to a calcitic skeleton when the water goes calcitic. I keep my aragonite. Okay? Now this isn't an absolute. It's a general trend that it appears to be fairly robust. Does that make sense to everybody? Okay. Now phosphate, people say, well, phosphate was really high, so that's why you get phosphate here. But as we'll hear from Philippe, vertebrates evolve phosphatic skeletons much later. Phosphate doesn't seem to, nobody knows why anybody uses phosphate. It's expensive. Why use it? No clue. And again, with silica, silica is again tough because we should have all kinds of things from the late Precambrian that are using silica, and we simply don't. So we got one trend here. The rest of it appears to be ripe for future research, basically. Okay. Now, in the previous talk, we heard about polyphyly. Right? What is polyphyly?
Ability spread over several phyla.
Yeah, basically it's convergent evolution. When we talk about a trait that's polyphyletic, what we mean is that a trait evolved convergently, at least 2 times independently. Okay? Saber-toothed tigers, for example, is a polyphyletic character. For some reason, tigers are going nuts with their teeth in the Pleistocene. You see them in Australia, you see them in California, nobody knows why. Polyphyletic. Okay? Now, this is a phylogeny of animals. There's our deuterostomes, the ictiozoans, the Lophotrochozoans. Okay? Shown here are some sponges and the cnidarians. Mineralogy, again, shown in colors, carbonate in blue, phosphate in green, silica in red. Do you agree that skeletons are polyphyletic? Yeah? I'm putting you on the spot here. Yeah? If they were monophyletic, what would you expect the pattern to see? Turn it and switch it around in your head. If the skeletons were monophyletic, what would you expect to see? Oh, you do know. If I said hair is monophyletic, and you looked across a whole bunch of mammals, what do you expect to find? Hair, huh? Okay. If your skeletons were monophyletic, and I looked all across the animals, what would you expect to find? Everybody would have skeletons, wouldn't you? And the fact that you see lots of lineages that are black, which means they don't have skeletons, and different compositions of the minerals suggests that maybe animals are multiple, invented skeletons multiple times independently. Who here would agree with polyphyly? Come on, you got to take a stand. We're doing science. We don't sit on fences, people. Polyphyly, raise your hands. Okay. 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 Monophyly. Okay. You are the. It looks like you might be right. Why monophyly? Oh, nobody cares what I think. We care what you think, right? What do I know? I don't know anything. Do you think monophyly? Here's the question, though. Who cares what we think? How do you test it? Right? Isn't that what's important? How do you test between these two hypotheses? I have a pattern here. Is that evolution? It's just a pattern, isn't it? We need to be able to get in there and test these hypotheses. You want to say that it's monophyletic? Fabulous. Refute it. What's your experiment? What's the experiment? Now, we heard in a previous talk, sorry, I don't remember your name, I apologize, that he had some interesting ways of thinking about testing the monophyly versus paraphyly of magnetosomes in bacteria, whether this last common ancestor had this ability, didn't he? He took you through some of these. What did he do? If animal skeletons are homologous, what would you predict? What did you predict? If magnetous bacteria, magnetosomes, are actually in a last common ancestor, what would you have predicted?
In this case, I would predict that they are, if it's monophyletic, they have the same genetics.
Isn't that a good prediction, right? It doesn't prove it, right? We don't prove anything in science. We're not doing math here. But it's a hell of a good prediction. If the last common ancestor of animals made skeletons, then one would predict that they probably use at least some of the same genetic architecture, wouldn't they? Okay, let's take a look. All right, what we're going to do now is we're going to switch over from the geologic record. There's a beer in it for anyone who can tell me what this guy is. Or does it have to be wine? Beer? Is beer okay? Beer's good. French make fabulous beer. It's amazing. Anybody know?
Critter. Critter.
It's a critter. Actually, we can put a name on this critter. We can even classify it as a bryozoan, the moss animal. Okay? They have the little lophophores. They're part of the Lophotrochozoa. And this is a little critter called Archimedes. And it's this corkscrew. And why I love the corkscrew fossil is I can take a strand of DNA and wrap it around because it's got the same pitch. Isn't that cool? To emphasize that if you're a paleontologist, people often assume you're out hitting rocks, collecting fossils. No, not necessarily. What you're interested in is the history of life. And you're going to use any tools at your disposal to get at the history of life. And paleontologists, to my chagrin, have ignored one half of the fossil record and focused on the other half. They generally look at this. They tend to ignore this. It's a different department, different set of tools and training. No, damn it. You have fossils in your DNA just like we have fossils in rocks. So let's go to the fossil record, the genetic fossil record, to see what does it tell us about the origin of skeletons. Now, this is a deliberately complicated slide, okay? So feel free to throw things. I deserve it. I did it on purpose, okay? Echinoderms. Here's all kinds of different echinoderms. This is just a small smidgen of echinoderm morphology. Here's that carpoid I talked to you about. Here's the Goggia with the 5 arms. There's a starfish, a crinoid. That's a sea urchin. And they all make their skeletons out of stereome. It's calcitic stereome. Would you expect them to share the same genes to make the same skeleton as a monophyletic group of animals that no one else does? Did you expect that? Yeah, I mean, it seems to make sense. I've got a lineage of organisms that's monophyletic, and all the living forms make the same kind of skeleton. And they've been doing it since the Cambrian. One then might predict that I bet they use some of the same genes. Because if they don't, imagine it was all different. It's like, wait, I guess this isn't going to work, right? So sure enough, if you look at how echinoderms are making their skeleton, we see that there's several hierarchical layers to making a skeleton in terms of the genes. We use regulatory genes. Who can name a regulatory gene? What do I mean by a regulatory gene?
The genes that regulate the biomineralization.
But don't just read. Give me like an example. What do we mean by a regulatory gene? Give me an example or a kind of gene that does this. Even in your bacteria. Oh, you know, you may not know your MAM genes, is that right, ma'am?
Yeah, but we don't know exactly the mechanism.
You know have transcription factors though, don't you?
Yes, of course.
Yeah, we have transcription factors. What does a transcription factor do? Doesn't a transcription factor regulate the transcription of other genes? That's an example of a regulatory gene, or a cell signaling. I bet your multicellular bacteria are communicating, aren't they? One would predict that they're actually communicating, they're passing molecules, and they're talking to one another, aren't they? That's another type of regulation. All your cells are talking to one another. Right? My heart cell knows it's a heart cell because it knows where it sits in space and time. And one of the ways it knows that is because of all the transcription factors. There's turned on genes and turned off genes, but it's also talking. It says, hey, I'm a heart cell. Oh, I'm an eye cell. Hey, that's what cells do. That's how development works. Now, shown on here, this crazy complicated picture here is from Eric Davidson's lab. Eric Davidson is one of the great developmental biologists of the 20th century. And him and his army of researchers have basically figured out gene by gene and cell by cell what the hell's going on. It's an amazing feat. We start from the top and we move our way down to the bottom, both in absolute time and in developmental time. Okay? So development moves forward. We start with a fertilized egg at the top and we work our way downward. All of these molecules you'll see in here are regulatory molecules, and we have all of them. Right? There's no difference between what you see for a sea urchin and what you see for us. We have ETS, we have Wnt8, we have ubiquitin, we've got, what else we got here? FOXO, FOXB, FOXN2/3, delta. We use Notch for signaling. It's all the same. It's not till we get to the bottom, and these cells right here labeled SKEL, probably short for skeleton. That's right, skeleton. They're labeled here in blue through an in situ hybridization. So we're actually staining RNA derived from this gene here called MSP130. And these cells go on to secrete the calcite that makes the larval skeleton of a Pluteus larvae. Okay? So all this information, all this wiring eventually leads to skeletogenic cells expressing things like SM27, SM50, MSP130, which at the time, we classify these as biomineralization genes, which are echinoderm-specific genes. So in other words, you go in, you look early, you see all the genes everywhere across animals, but you look late, the cells that actually do the skeleton, and those are echinoderm-specific. Okay? And the prediction is that all living echinoderms will have the same basic machinery built into it because they all make stereo. Okay? Does that make sense? Okay? There's a quiz on this. Just kidding. I want to be invited back to your lovely city. Okay. Here's a gene called spherulin. It's derived from a sponge. The sponge is really fascinating. This is from the work of Dan Jackson and his colleagues. It's a demosponge, so it makes a glass skeleton, but it also makes spheres of calcium carbonate. It's got a calcite skeleton and a glass skeleton. Okay? This is in situ of the cells that are actually secreting the calcium carbonate, and they're expressing this gene called spherulin. Okay? This is a phylogenetic tree of everything known to have spherulin. We've got, and I'm terrible with, that is delta? Is that right? I've never joined a fraternity. I don't know how else one learns this stuff without actually being learned. Delta and gamma, okay. And we heard about these, didn't we? These are proteobacteria, okay? And then two sponges. So you don't have this gene. What do you think? Where did the gene come from?
Well, first I thought that sponges were boring, and now you're telling us that they're very fascinating. From the same difference between bacteria and sponges?
So about, I don't know what it is, 80% of the biomass of the sponge is bacteria. Basically, they're bacteria and a little animal, right? They're like, they have a little bit of animal added to it for flavor or something, but basically they're bacteria. Now this gene, all right, sits here. It's got an intron with a splice site, branch point, urididation signal. Those of you who know eukaryotic genes, like, wait a second, that's a eukaryotic gene. Do bacteria have introns? Splice sites? No. Okay? So this gene, even though it exists in both of these domains, and in the sponge it has a eukaryotic organization, but in the bacteria it does not. It's into an operon, just like the genes that he was talking about. Where did the gene come from? Now, if I heard you properly, you thought it was from the bacteria, maybe horizontally transferred over to the sponge? Why can't it go in the other direction? How do we know directionality here? Can't it go this way as easily as it can go that way? Go ahead.
I would think that Proteobacteria appeared before Metazoan, so I would think it would go that way.
Did everybody hear that? She just told time, didn't she? Proteobacteria came before sponges. Now, there's a caveat we have to add into this, though, is that right now everything's just living, right? How do you know time based on these data? You do, because you know, as we saw earlier, that the split between this and this encompasses all proteobacteria. And all proteobacteria include the mitochondrion living in our cell. So that split has to be older than the origin of eukaryotes. Anytime you look at a tree, you can tell relative time. Study the tree node by node until you can familiarize yourself or ground yourself. Say, wait a second, I know roughly when that occurred. This gene is probably greater than 2.5 billion years in age. It's at least as old as 1.6 billion, probably much older. So that's, because it can go either way. In a eukaryote, it looks like a eukaryotic gene. In a bacteria, it looks like a bacterial gene. Okay? Find your nodes, tell some time. Then you get polarity. Let's go back to MSP 130. Now, when we wrote that paper in 2006, as far as I know, maybe I didn't get on Web of Science that day. Hell, I don't know. It was only found in echinoderms. Chuck Ettinson, a worker who studies these cells in sea urchins, published this phylogenetic tree of MSP 130 genes. Echinoderms are shown here at the top, and there's multiple copies. We're not going to worry about the multiple copies, okay? But all echinoderms basically have more than one copy of MSP 130. There's their nearest relative, the hemichordate. Hemichordates don't make skeletons, but they have MSP 130. That's kind of interesting. Here's us as a close relative, Amphioxus. We don't have MSP 130, but a close relative of ours. And again, an animal that doesn't make a skeleton has MSP 130. Shown here are the only other animals known to have MSP 130, mollusks. So here's oyster, Crassatria, and Lloydia, snail shell. Okay? Volvox and Chlamydomonas. Who knows what Volvox and Chlamydomonas are? What are they? No? Green algae. Okay, so these are green algae. So these are, this last common ancestor of Volvox and sea urchin is probably the last common ancestor of eukaryotes. So we've gone way back in time now. Okay? Plants don't appear to have it, but their green algal relatives appear to have it. And shown here now are a whole bunch of bacteria. Okay? So the green dot is all the bacteria. Now if we put a line there and a node, that's Luca. What's Luca? The last universal common ancestor. So based on this tree, it would appear, shockingly enough, that this goes back to the mother of all life on Earth. Chuck writes at the end of his paper this. I propose that MSP 130 genes were introduced into metazoan lineages via multiple independent horizontal gene transfer events. What do you think of the hypothesis? What do you think? Exactly. That's exactly the right answer, right? It's interesting. You buy it? Let's go back to his example, okay? He was throwing around horizontal transfer, right, across all these different bacteria. Now he put up a very key slide, didn't he? He showed two trees, one derived from his genes and one derived from a universal marker 16S. And what were those two trees? What did they show? Showed concordance, didn't they? They were the same topology. Is that consistent with horizontal gene transfer, or is it consistent with vertical transmission through evolutionary time? If I said your nearest relative was a sequoia tree, you just happen to share hair and fingernails, eyes with chimpanzees, right? What would you think of that? Would you say, whoa, that's shocking, that's interesting, right? You say, well, wait a second, I can test that, and I look at trees, and I sequence genes, and I find, wait a second, geez, we keep coming out really close to chimpanzees and not sequoia trees, right? Do we have bacteria interspersed throughout the metazoan side of this tree? Or are animals, metazoa, monophyletic? What do you think? You're lost? Well, let's start again. We're going to start from a single critter. I'm a critter, and I have MSP 130. And you want to know about where that gene came from. So we sequence a near relative, right? Not sequoia, we're going to sequence chimpanzee, okay? Chimpanzee has that gene. And I keep sequencing and sequencing and sequencing, and I get to a plant, and the plant doesn't have it. The bacteria doesn't have it, and the archaea doesn't have it. Only the animals have it. You say, oh, I bet it's a monophyletic gene, and it arose one time in the early history of animals and been shared across all the descendants, right? That's what you would probably infer from that, right? Is that what's shown here? I have one animal, Helicidaris erythrogramma, and I sequence all these other animals and do a phylogeny, and bam, I come down to a single animal. Isn't that consistent with monophyly?
So these are all the organisms that where they look for it?
Yes, they looked everywhere. This is what has it.
In eukaryotes.
Hmm?
In eukaryotes. So
Anywhere that has a sequenced genome.
independent acquisitions possibly there.
Okay, so let's imagine that there's two independent acquisitions.
One for the chlorophytes, greenhouse, another for the animals. Likely, but we don't see that in the tree.
We don't see it in the tree. What do we see in the tree? What do we see in the tree? Remember, the tree's the data. Chuck looks at the tree, writes the paper, suggests horizontal gene transfer. But the only data in the paper is the tree. Why is he even suggesting horizontal gene transfer? You guys actually basically kind of formulate it. Is it because we don't find it very many places? Right? We find it in mollusks, but I don't find it in annelids, I don't find it in flatworms, I don't find it in arthropods, I don't find it in nematodes, I don't find it in any other animal. In fact, he goes on to postulate that each hemichordate, amphioxus, and sea urchin is an independent horizontal gene transfer event. Are you happy? If it's coming from bacterium, what is the prediction, though, with the tree?
That each animal branches close to each different bacteria who were responsible for the transfer. So you see monophyly there of animals.
Yeah, we're going to root it here, okay? Oh. Can we cut that from the film? Is that a little splice right there? We're going to root it here, okay? So we've got a monophyletic bacteria, right? We've got a monophyletic green alga, okay? And we've got a monophyletic metazoa. What we'll do is we'll use bacteria to root the tree, so we're reading evolution in this direction, right? If you saw bacteria, a bacterium here, a bacterium here, a bacterium here, a bacterium here, that's exactly what you would expect. And that's how we realize as a group, as scientists, the phenomenon of horizontal gene transfer. But here we just have a gene that seems to be present in a monophyletic group, what we call metazoa. Does this seem a little weird?
Could it be a mutation?
Well, everything mutates, right? I'm mutating right now. Is this phylogeny consistent with this gene being present in the last common ancestor of at least echinoderms and mollusks? Yes. We could have tremendous amount of loss. I think that's what we were seeing with the magnetosome tree. We have a tremendous amount of loss, don't we? If your tree is right and you don't have horizontal gene transfer, the alternative is there's a tremendous amount of loss, like in mitochondria, for example, right? What if we're just looking at it's just a bunch of loss? So that animals that don't have skeletons lose them, and the animals that retain skeletons, at least a couple of them, have this gene. Suggesting potentially, throw it out there, that maybe the last common ancestor actually had a skeleton. And things like Nemernians and flatworms simply lost skeletons, and so they lost their MSP130 gene. Never, ever, ever forget about loss. It's the one idea, for some reason, that we just don't consider very often. Bacteria lost their introns. What? I think that's probably true. Bacteria lost introns. We didn't gain them as eukaryotes. Bacteria lost them. And I know it's a consideration from the biochemistry of it all that suggests that splicing is primitive. So let's take a look at this. Here's another gene here called astrosclerin. It's expressed in the same cells we talked about earlier. Right? So this is the sponge. Here it's expressed in the skeletogenic cells of the sea urchin embryo. Here is a tree. We have it. Guess where we express it? Hard parts, cells that secrete bone. Okay? Sea urchins have it. Sponges have it. Cnidarians have it. Annelids have it. It's carbonic anhydrase. Last common ancestor have skeletons? How many genes do I need to show you that are conserved between various animals before you would actually say, gee, maybe skeletons are homologous between sponges and echinoderms? How many genes would it take? 2, 5, 10, 20, 100? For some developmental biologists, as Philippe was discussing earlier, it's just one. You see a gene, oh, bam, they're homologous. Right? There is no known way, is there? It all feeds into our psychology and what we need to be convinced. What were our prior assumptions about this phenomenon? I assume they evolved independently. You're going to have to show me a lot of evidence to show me I'm wrong. But if I think that if I had the assumption or I was taught in school that these are homologous, it's not going to take very much. The only way to really test it is we have to leave that system and try to test it another way. Something totally independent, or at least as independent as we can get. We can't just keep looking at genes, more and more genes, because it's just going to, you're just going to be convinced of what you already know. So let's change the way we look at the phenomenon. Come back to this question, and let's just find out when animals evolved. Because at the moment, all we've talked about are when animals make their first appearance in the fossil record. Is that actually when animals evolved? To some paleontologists, yes, of course, because otherwise I'd find the fossils. Makes sense. To other people, it's like, wait a second, no. What if skeletons in large size just happened multiple times independently? Maybe animals have a very deep history. And if they do, and we're not finding skeletons in the Precambrian, then that would be very consistent with polyphyly. If, however, animals themselves have evolved, say, in the early Cambrian, boy, maybe it is possible that this shared genetic machinery indicates commonality of skeleton. This is Bruce Runniger, one of my intellectual heroes, founder of molecular biology, and in particular was the first to really use a molecular clock to date the origin of animals, to get at whether we're looking at the rise of skeletons, say, or the rise of animals. It's the only way to actually tease it apart. You've got to look at the other record. He did a very interesting analysis with hemoglobin and came to the shocking conclusion that the last common ancestor of us and a fruit fly lived 1 billion years ago. A long time, right? 500 million years before they show up in the fossil record. Let's take a look at how a molecular clock works. Now, this isn't biomineralization, but I want to be able to show you this because I think it's an important tool to be able to get at and think about the origins of anything, skeletons, what have you. We're going to go down to the beach. We're going to get some critters. You have critters living on your shores here. I assume some of them are sea urchins. Okay? We're going to grab some sea urchins because they have a really good fossil record. I'll show you why that's important in just a second. These are the ones that we would get. This is the west coast of the United States. S. purpuratus. Eucederis lives in Florida. Fruit flies. Mosquitoes, I assume you have them. Okay? Yeah. And a cnidarian. Okay? A little thing called nematostella. It's like a little sea anemone. This is the sequence of a gene called aldolase. Everybody has it. We use it as part of glycolysis. Okay? For those of you that don't remember your biochemistry, it sits right in there. It's part of splitting up the carbon. Okay? We don't really care what it does. What's important is that everybody has it. So I can go into my lab and actually get it from whatever I bring in, from whatever I find at the seashore. Now, that's two sea urchins. You'll notice the boxes indicate differences in the amino acid sequence. Someone said things mutate. This is mutations. Okay? There's a mutation here. I've got a methionine. In Eucederis, I have an isoleucine and S. purpuratus. Okay? Everything's mutating. Now, this is a phylogenetic tree of those data. Here's the two sea urchins. Here's the two flies. There's nematostella, the deeply diverged cnidarian. Okay? I'm going to calculate how many differences there are between these two genes. Just count it up. How many differences are there? Okay? Those numbers of differences tell me the genetic distance then, if you will, between these two animals. If I can relate that to the amount of time since they last shared a common ancestor, I can calculate a rate. From the fossil record, we think sea urchins share our last common ancestor about 250 million years ago. So by taking this amount of genetic distance over that time allows me to calculate a rate that I can then use to ask when did flies diverge from one another or even when sea urchins diverged from flies. The molecular clock is very simple in principle. See how many mutations they are, calibrate it to the known fossil record, make an inference when they last share a common ancestor where I don't have fossils. Okay? It's just simply an extrapolation. Questions? Are we good on that? Okay. We've been doing this for a long time in my lab. This is actually, most of this work was done by undergraduates. This is the latest analysis that we've published. There's about 120 taxa in this, or 118, okay? It's 7 different housekeeping genes, like the glycolytic genes I showed you. We concatenate them together. The white circles are calibration points to the known fossil record, okay? I don't want to bore you with the details of how we did it. What we're trying to get at is to relate this now to time. Shown in blue is the cryogenian. Do you have the word cryos in French words? Yeah. The name comes from the snowball Earths, okay? So Earth froze over twice, at least twice, during this time period. Shown here in brown is the Ediacaran that we talked about, and shown here in green is the Cambrian. We estimate that the last common ancestor of us and a glass sponge, what you take a bath with, the sponge you use to clean your body, that last common ancestor lived about 773 million years ago. Shown at the bottom here are just the errors, okay? The last common ancestor of us and a fruit fly, about 668 million years ago, well before the Cambrian, almost 150 years before the Cambrian explosion. Does that seem surprising to you? Are you surprised? Oh, sorry, bilaterans, anything with bilateral symmetry. So us and fruit flies, for example, us and a worm, us and a clam. It's interesting. Now, what do you work on?
I'm a geochemist.
You're a geochemist.
Yes.
You could care less.
No, it's
Well, I mean, professionally,
true,
but for professionally.
no, not at all actually, because I'm a geochemist and I work on larvae a lot. And
Ooh.
actually, it's where it gets very
Oh, so now, but there's a metazoan connection. So if you're just doing a geochemist, the geochemists I know, they're chemists, right? And they're like, well, whatever. Because they got no assumptions about what the fossil record should look like, right? But if you work on animals, it's like, wait a second, I got some assumptions I'm going to build in this. Like, how can you have animals that old and I don't have any fossils of them? But you work on larvae.
Yeah,
Larvae can be there all over the place. You wouldn't find anything, right? Yeah, doesn't bother her. Again, what's in your mind dictates how you view these data. I set out to refute the idea of a deep molecular clock. I came to realize that, geez, I can't refute it. It actually seems fairly robust. But it's not as deep as people once thought. How many of you have heard molecular clocks are garbage? Yeah. What did you hear? That the rate can vary, right? Now, I made an assumption here, right? We don't actually make that assumption in the analysis I showed you, but you're exactly right. Rates can vary, right? What about you guys? Exactly the same, okay? I can actually show you, even if you go back and look at my original papers, I actually show you the rate doesn't vary in these molecules, which was really shocking, but it actually doesn't. This is, for a paleontologist, this is why most paleo people thought it was all garbage. This is sterium again, hard skeletal calcite, right? Can't miss it. Last common ancestor of starfish and a sea urchin, according to this molecular clock study, 581 million years. Last common ancestor of all living echinoderms, 730 million years. Possible? How are we doing for time? Are we okay? Okay. Is it possible? Can I ask a paleontologist? Is that possible? That you'd be missing sterium for over 200 million years? 200. Last common ancestor of a crinoid and a sea urchin, 730. Huh? 200 meters. That's like 220 maybe. It depends if you know. 215. I mean, 195. It's a lot. What do you think, though? You're a paleontologist.
The sterium is there much long before. In the Silurian, there is sterium.
Well, remember, though, we're 730 million years ago is the last common ancestor of animals that have sterium. So we should have records of sterium from 730 million years all the way to the recent. That would be like me saying, what if you had heterostrachan material at 730? Would you be, oh, wow, that's really fabulous?
Yeah.
Impossible. That's the reaction from an eminent paleontologist. Impossible. Which is why paleontologists said, the hell with this stuff. This doesn't work. Right? Well, what I want to show you is that mine works, because we get good dates for things where we know where the skeletons evolved. This is the last common ancestors of all the living phyla that we have. Okay? We date the last common ancestor of echinoderms at about 510 million years. Okay? Sterium shows up about 515. In other words, we don't have a sterium gap. Right? When we date the minimum, we get it into the Cambrian. Same with brachiopods. Brachiopods show up about 520. That's also when we date the origin of what we call crown brute, the last common ancestor of all the living brachs, at 520. Okay? Arthropods, about 525. Basically about when trilobites show up. So in other words, this clock appears to be fairly robust where we can tie it in to the known fossil record. Okay? We don't have these 100 million year gaps that people like Philippe would just say, your method is not working, maybe because you have a rate difference. When we do it, and where we can test it repeatedly, we've tried multiple times, it appears to be fairly robust. And my point is that if these points are good, then maybe that point needs to be taken seriously. That the last common ancestor of a sea urchin and a mollusk is about 680 million years ago. Okay? This guy right there, 680. Did that animal have skeletons?
Which one?
The last common ancestor of sea urchins and mollusks. Did that animal that lived 668 million years ago, according to my molecular clock, have skeletons? Mm-hmm. Yes, we're the ones that published that. I took it out for time. Okay? We think there's a taphonomic reason why we're actually missing the silica. Okay? And people are actually working on recent finds where they think they're actually finding the silica. Okay? But I kind of want to come back to this to make sure we understand how we would know that we've got, say, a sponge spicule gap. Does that make sense? I'm not in any way deferring your question. I'm just saying, yes, you're right. We published a paper called Where's the Glass? It's a 200 million year gap in the fossil record. We don't know why. We think it's a taphonomic reason why we're missing the silica. Okay? Basically, because if you don't have any clays, you don't have any alumina, we just can't keep it around. Okay? It doesn't change until you get into the Cambrian. It's a clear artifact. But this, right, this actually gets at the main question. If that last common ancestor lived 668 and it's not silica, it's calcium carbonate, would you expect there to be skeletons? Remember, you have multiple records now you have to integrate. We have a fossilization record, we have a phylogenetic record, and we have a molecular record. Put it all together, what does that tell you? It's important to know there is no right answer, right? We don't know. But putting these three records together in your mind gives you a framework to then test against, to try and refute it. To me, and again, what I think doesn't matter, but to me what this tells us is that these genes are present in these ancestors. They're being recruited in multiple times to make skeletons. Sometimes we have innovations like a gene from bacteria into sponges, but a lot of the times I'm just using whatever I can in my toolkit to get at the formation of skeletons in the early Cambrian. But it's possible skeletons are homologous, right? It's just not consistent with our understanding of the known fossil record, okay? So whenever you're looking at these trees, you've got to find the weak point. How can I refute this? Where's the weakness at? How can I get at this? I ran out of time. I just wanted to, let me just summarize the last few slides. Basically what this all boils down to me, the independent rise of skeletons reflects the inventions of marine ecology. That is the Cambrian explosion. We go from a two-dimensional to a three-dimensional ecosystem right as we cross the boundary. Animals figure out how to get up into the water column. The same time they figure out how to get in to the sediments. The same time they figure out how to formulate skeletons. It's all being driven basically by predation. I'll show you one final slide. I'll skip this because I know I'm running out of time. There's Claudina. That's a borehole. Right? That hole you probably have noticed. Right? The very first skeleton we have in the fossil record has traces of predation on it by something that was drilling into it to try and eat it. Right? So when we think about the formation of skeletons, there's nothing really special about it in a sense. It's part of a much larger ecosystem framework. This changing of a two-dimensional to a three-dimensional ecosystem that's largely being driven by carnivory in the presence for the first time in Earth's history of enough oxygen to be able to support deep food webs. All right? So the whole Earth is basically changing at this time. And with that, I'll stop. Thank you very much.
Thank you.